Now we're going to focus on talking about how eukaryotic cells go through this process. Eukaryotic cells have specific numbers of chromosomes that they rely on, and they need to make sure that all the chromosomes are exactly where they belong. This means it's a little more complicated than just cutting yourself in half. Before we can really talk about how chromosomes behave, we need to talk a little bit about their structure. So we know that chromosomes are made up of DNA, and that DNA is a double helix made of nucleotides. What you might not have known is that DNA is actually more than that. We can kind of think about DNA like going to the yarn store. If you have a ball of yarn, it's a lot easier to carry that yarn if it's all wound up into a ball. It's a lot harder to carry it if it's all unwound. Your DNA isn't much different. Your DNA is going to be wrapped around proteins called histones, and those histones will be wrapped into coils. Those coils will further coil into supercoils, and these supercoiled materials are what we call chromatin. So DNA itself is just the nucleotide double helix, but chromatin is packed DNA wrapped around histones. We can actually see this when we look at condensed chromosomes, things that are tightly wrapped. These are Drosophila chromosomes. Notice that they are linear. Prokaryotic chromosomes are circular. The banding pattern of these will also be important later. If we were to look at them in the context of a nucleus, this is what they would look like. Now, do you know how many chromosomes a human being has? It's okay if you don't. The answer is 46. Human beings have 23 pairs of chromosomes. You can see this in the karyotype right here. A karyotype is just where chromosomes have been laid out next to each other. This particular karyotype belongs to a male. We can tell by looking at the 23rd chromosome, where there's one X chromosome and one Y chromosome. Now, all of these chromosomes come in pairs. There's a special name for that. These are homologous pairs. Homologous pairs have the same information on them. They might encode the color of your eyes, for example, but they're not identical. One might say that your eyes should be green. The other might say that your eyes should be brown. The reason you have two is because one of each will come from one of your parents. So in this example, the blue are coming from your father. They're the paternal copy. And the purple are coming from your mother, the maternal copy. Before the cell can divide, it first has to replicate its DNA. When we think about chromosomes, we often think about them in this X configuration. But chromosomes normally are linear. In the center of the chromosome is what we call a centromere. When replication happens, this copy of the chromosome will still be attached to the centromere. These identical copies are called sister chromatids. Sister chromatids are identical, whereas homologous chromosomes are not. So the basic idea here is that we'll start with chromosomes, which are DNA molecules. We'll duplicate each chromosome to form sister chromatids. They're going to look like our actual little X connected by the centromere. And then we're going to separate these identical sister chromatids into each of the two daughter cells. Now, it's not quite as simple as that. There are a lot of phases to mitosis. The largest and perhaps most important phase is interphase. Interphase itself is also made up of multiple phases. G1, the first gap, S phase, where DNA synthesis occurs, and G2, the second gap. The amount of time that a cell spends in mitosis is actually quite small, and this is called the mitotic or M phase. M phase itself is also made up of multiple phases, so let's take a closer look at what's happening during each one. Cells spend the majority of their time in interphase. In interphase, DNA is decondensed and fills the entire nucleus. Individual chromosomes cannot be seen. You can see in the nucleus above that it is entirely blue with no discernible or noticeable chromosomes. At the same time, centrosomes exist but are not yet growing to create a mitotic spindle. The centrosome is a special organelle that only exists in animal cells. When we talk about mitosis and meiosis, we'll be focusing primarily on these types of cells. The centrosome is important because it is made up of microtubules that are going to guide where chromosomes will go during the process of mitosis. During interphase is also when the cell is going to replicate its DNA. This happens during S phase and is critical for mitosis to be successful. After interphase, the first phase of M phase, or mitosis proper, is prophase. During prophase, the centrosomes will grow, forming the mitotic spindle, which is a connection of microtubules. Chromosomes will start to condense and become visible within the nucleus. If you look closely at the cell above, you can see that instead of being uniformly blue, there are little squiggles inside. You can also see that the mitotic spindle is starting to form, those long, thin green projections that are coming from where the centrosomes are located. Now, your textbook divides prophase into two phases, early prophase and prometaphase. During prometaphase, this mitotic spindle will start associating with kinetochores, which are located on the chromosome, and the nuclear envelope, which keeps the chromosomes inside the nucleus, will break down. For our purposes, we are going to combine prophase and prometaphase. So for us, in prophase, chromosomes condense, the mitotic spindle forms and associates with chromosomes, and the nuclear envelope breaks down. The next phase is perhaps the most iconic phase, and that is metaphase. During metaphase, the mitotic spindle applies pressure to the chromosomes, which causes them to line up in the center of the cell. You can think about this like an object with strings tied to it. If you hold that object, you can suspend it evenly between your hands by the strings. That's what's happening here. Metaphase is relatively short. As soon as all the chromosomes are lined up properly, it will end. The following phase is even shorter, and that's anaphase. During anaphase, the centromeres, which hold the two sister chromatids together, are going to break down, allowing the chromosome to separate and the two sister chromatids to start to move toward their sides of the cell. Once they reach their side of the cell, that will be telophase. And when that happens, the cell will begin to divide in what is known as cytokinesis. Now, cytokinesis occurs differently in plant and animal cells. In animal cells, a cleavage furrow will form. That cleavage furrow looks kind of like a ring around the cell. You can think of it like a drawstring bag. As you pull the ring, or the string, it causes the cell to pinch off until the two cells separate. If you're having trouble remembering about the cleavage furrow, you can remember that it looks kind of like an old person's butt. That's what I used to think about it. Now, if we think about how plant cells might work, how are plant cells different from animal cells? Well, when it comes to dividing, the biggest difference is that they have a cell wall. The cell wall provides a problem. If you were to tie a string around a cell wall and pull, you wouldn't be able to pinch the cell apart. Instead, you have to create a divider from the inside. So what's going to happen is that vesicles containing extra wall material are going to line up in the center of the cell. They will then fuse to create what we call the cell plate, and the cell plate will move outwards until it divides the two cells in half. So animal cells divide from the outside inward using a cleavage furrow, and plant cells divide from the inside outward using a cell plate. If you want to watch cells divide, I've included two different videos that you can watch. It's worthwhile to try and see if you can recognize all of the major phases. It's going to be very difficult to tell the difference between telophase and cytokinesis because those things happen roughly at the same time, but it's still good practice. Now we're going to talk a little bit about how we regulate cell division because the regulation of cell division is extremely important. The first thing to keep in mind is that cells do not randomly divide. What normally occurs is that when you grow cells, for example in cell culture, The cells will divide until they reach some signal that tells them to stop. In the case of your skin cells, for example, they will continue to divide until they have created a single layer of cells. If you were to remove some cells, they would then divide to fill that space. The only time that cells will randomly divide and ignore these kinds of signals is when there's something wrong with them. This is normally the case in cancer cells. Cancer cells are either unable to detect the stop message or they have an overactive divide message. Either way, they are behaving abnormally. Generally, there are three major places where cells will check to see whether or not cell division should occur. These are the G1 checkpoint, the G2 checkpoint, and the M checkpoint. If we think about what's going on at each of these time points, we can get a good idea of why these checkpoints exist. For example, the G1 checkpoint takes place during the G gap phase, but before DNA replication has occurred. This is a good time for the cell to check, have I gotten any messages that tell me I should prepare to divide? If I have, I should replicate my DNA. If I haven't, I should stay in G1. Once the cell has replicated its DNA, it needs to decide whether or not it should enter M phase. This checkpoint at G2 determines whether or not the cell should continue with the process. Has it gotten a signal to tell it to actually divide? Some cells replicate their DNA because they need extra chromosomes, but they never actually divide. The other thing the G2 checkpoint is for is to make sure that DNA replication has occurred properly. If it hasn't, the cell can abort going into M phase, take apart the synthesized DNA, and return to G1. The last checkpoint is at the end of M phase, before cytokinesis occurs. This checkpoint primarily exists for the cell to check and make sure that cell division has occurred properly. If it hasn't, it can try again. Generally, the messages that cells will use to determine whether or not they should pass through a checkpoint will come from one of two places. They will either come from outside the plasma membrane, in which case we call this a signal transduction pathway, one where a receptor that comes from outside interacts with a membrane protein, which interacts with other proteins in the cytoplasm. Or they might come from inside. An example of one of these would be growth factor. When your cells are exposed to growth factor, this tells them that you are in the process of growing and that they need to divide. This allows them to bypass the G1 checkpoint and start to replicate their DNA. These kinds of signals rely on a wide number of proteins, which we call relay proteins, to make sure that they occur properly. This is why it's so easy to have problems with growth, and why it's one of the most common types of developmental abnormalities.